Hi guys, so we're going to start the final unit in our genetics course. So we're going to start with chapter 12, and this is all about the genetic code and transcription. Okay, so I'm going to share my screen with you guys and my PowerPoint. Here we go, so genetic code and transcription. So we know all about DNA, right, and how that basically encodes it's a program, it's the code that encodes the rest of like our cellular features, the things that actually make us work, right? So now we're going to look into how DNA is going to be made and expressed and read into a protein. So the DNA doesn't get directly made into a protein, though initially they actually thought this was the case, but that does not happen. It has a little intermediate step, and it's made into something like an intermediate molecule before it gets fully made into a protein. So we have our DNA that is made into mRNA, right? And then from that, our mRNA is then read and made into a protein. So I'm going to keep up my PowerPoint screen right there, but I just want to, real quick, we're going to have our DNA right here. If this is the gene, remember we have our entire genome is humongous. Only like a certain gene, there's actually high regulation in what gets transcribed and what actually gets expressed. So gene A is going to be made ultimately into protein A, this little weird ball right there, okay? So when this happens, our DNA, first off, has the actual gene itself, the part that's being expressed. So this whole process, when we go from DNA to protein, that's known as expression, okay? Our DNA separates, and then one of our DNA strands acts as a template for our transcription machinery to start reading it and making the complementary RNA strand. So here we have the mRNA strand that was complementary to that template strand that we use from the DNA. And then this process is known as transcription. Okay, and then we go from mRNA to protein. This process is known as translation. One easy way to remember which one's which, transcription starts with this, like it has a C in it compared to translation, which has an L. C comes before L. Okay, so this is basically the central dogma of life, something that you guys know of very well. So now we're going to look into the actual genetic code, the code that we have on the mRNA, right, that we made from the DNA. It's basically, it's the same code, but just a little bit, like it's the language is a little bit different. Remember, mRNA and DNA both are still nucleic acids, still the nucleic acid language, but the nucleic acid language of RNA is a little bit more comprehensible to the protein synthesis machinery than the nucleic acid language of DNA. So what this nucleic acid language, right, it has a code, and this code is known as the genetic code. And so what's the genetic code? We're going to look into and kind of decipher and figure out how the genetic code from here, from nucleotides or ribonucleotides, so A, G, U, C, how that becomes, one of the 20 amino acids that we have out there. Okay. So the flow of information from gene to protein is based on the genetic code. It's a series of non-overlapping 3 nucleotide words. There's a couple of features of the genetic code. The first one is that it is linear. And so you start at the beginning of the mRNA, which is in linear format, and then you just basically read along until you get to the end. You read every single word, right, which makes one amino acid in a triplet. So basically 3 ribonucleotides are going to encode for 1 amino acid. So 3 ribonucleotides, that's equivalent to a triplet. Another name for this that's going to be used on forever, and I'm going to use it from now on, is known as a codon, right? So 1 codon is equal and encodes 1 amino acid. And this codon is composed of 3 ribonucleotides. The other thing about the genetic code is that it's unambiguous. So if you take 1 triplet or 1 codon, it's always going to equal that same amino acid. It's always going to encode the same amino acid. It's not going to encode another kind of amino acid or other 3 different amino acids. It's always going to encode 1. So if we actually look at the genetic code right here, which I have on my slides, it's beautiful. No, I do not expect you to remember this. The only ones I expect you to remember are the ones that are highlighted, and I'll get to them in just a second. So, for example, we have CAU, right? CAU, which is, if you see this, so it's in 3 ribonucleotides, and that encodes the amino acid HIS, so that's the amino acid histidine. And so if you look throughout the rest of the genetic code right here, you're never ever going to find another CAU, right? And so CAU is only ever going to actually encode, and only ever does encode, histidine. That's it. Yeah. Though even if the genetic code is unambiguous, right? So each triplet code or codon equals one amino acid. You'll notice though that many amino acids, that many triplet codes can actually be used to encode one amino acid. So for example, you can see here proline is actually encoded by four different codons, right? So each codon only ever encodes proline, right? But we can have many codons that encode that same amino acid proline. Leucine is encoded by 6 codons, right? So it could be vary. I think there's only tryptophan, and I forgot the other one. There's another one that is only ever has one codon that encodes it. Here's tryptophan. I thought there was another one, but I could be mistaken. Anyways, so sometimes, very rarely, do you have one codon equals one amino acid and that's it. No more codons equal that same amino acid. But usually you have many codons that can equal that same amino acid. However, it's always unambiguous, right? One codon isn't going to again, CAU is going to encode histidine, but it's never ever going to encode something like serine or proline. It's always going to encode histidine. So that's what it means by being unambiguous. It also has grammar to it. You have a start and a stop, right? So this is where those 4 highlighted codons that I mentioned before are important. You do have, yes, your mRNA is read in linear format, but so pretend this is your mRNA, right? And your protein synthesis machinery latches on, your translation machinery latches on. We'll get into what the translation machinery is later on, right, in another chapter. And it starts reading the mRNA. It doesn't start making the mRNA into a protein right at the beginning, right? It only does so once it arrives at a specific a specific codon. That codon is known as the start codon, is AUG. And that AUG codon is always going to encode methionine, right, which is a specific amino acid. So once it gets to AUG, that's when it starts actually reading the mRNA and making it into protein. So the AUG basically establishes the reading frame of the codons as well. And then it won't stop reading this mRNA until it gets to a stop codon. There's 3 stop codons. We have UAG, UGA, and UAA, right? I wanted to see if I remembered those before I looked at the genetic code. I think stop codons are awesome. I want my license plate to have one of the, I think UGA or UAG, I think UAG is one that most people know, to have that on my license plate. I think, and in my car, on my car, I think that'd be really funny. Okay. Anyways, so now we'll actually see how the genetic code was established, right? They had to figure out a lot of things. First off, they actually even had to figure out that we went from DNA to an intermediate like mRNA and then to proteins. Initially, they thought that DNA was expressed directly into protein at the beginning, and obviously that's not the case. And so they realized that there was something in the middle that had to happen. This was realized by Jacob and Monod in, I butchered their names, they're French, Francois and Jacques. In 1961, they postulated the existence of mRNA. So again, that when your gene is needs to be expressed, it basically separates, and then the RNA or the mRNA transcript basically is made based on the complementarity of the bases of the DNA in the template strand. So then the RNA transcript is made, and then that is what then gets translated to be made into a protein by the protein synthesis machinery. So that was basically the first step. The next step was actually figuring out how big the code was, right? So they were like, okay, we know that there's 4 ribonucleotides, A, U, G, C. There's 4 of them. But then how many do you actually need to code for an amino acid, right? How many do you actually need? So they were like, if we have a combination of 2, actually I'm gonna change my screen over, hold on. Okay. So if we had a combination of 2 of those 4 nucleotides, so it'd be, we know we have 4 nucleotides, and now we're putting them into 2 possible positions. so 4 to the power of 2, that gives us 16 possible combinations. 16 possible combination doesn't quite cut it because we know that we have 20 amino acids. So they were like, it can't possibly be read in groups of 2 because if that was the case, we wouldn't arrive at the amount of amino acids that we need in this world to function. So they're like, okay, how about if we have 3 possible like combination of a triplet, right? So we have 3 positions that like, you know, that are read, and that can be read and made into 1 amino acid. Okay, if that's the case, 4 to the power of 3, that gives us 64. Okay, that starts sounding like a really like a good number. It's a lot more than the 20 amino acids that we need, but that's better than 16 because 16 doesn't even quite make it to 20. And they're like, okay, so the last possibility is to arrange those 4 nucleotides into 4 positions. So that would be 4 to the power of 4. Guys, I'm sorry, I don't know what 4 to the power of 4 is. I gotta take out my calculator. Here it is. There we go. 256, way more than 20 amino acids. So with this, they basically like kind of hypothesized that the code was read in groups of 3, so in triplets, so in codons. And so this is basically what they hypothesized because 256 was way too much, right? And so this was basically the hypothesis they postulated. Then the experimental evidence came from Crick. They started seeing that codons must be read in a correct reading frame, right? So, oh, that's something I forgot to mention. Sorry, I'm so sorry. The other thing about the genetic code, wow, I completely skipped this, is that it is non-overlapping. So non-overlapping, I'm sorry, I skipped it. Non-overlapping means that when, oops, wrong camera. When your code is read, when your code is read, so say you have a code that goes like so, etc., it's read in a non-overlapping fashion. So your triplet is read. This is your first triplet, which is equal to 1 amino acid. Okay, this is your second triplet, which is equal to the second amino acid. Right? This is non-overlapping. If it was overlapping, this would be your first, then this would be your second, this would be your third, this would be your fourth. See how it's really confusing? That's not the case. It's non-overlapping, and therefore we have a specific reading frame. So once we get that start codon, which yes is AUG, remember, that start codon that starts the expression of the mRNA, then and only then are we going to start like establishing our reading frame. So we start for every 3 ribonucleotides that are encountered are equal to 1 amino acid, then the next 3, then the next 3, then the next 3. And that is basically the reading frame that we're talking about. The other thing I forgot to mention about the genetic code is that it is universal. So I'm sorry, I'm going back on my other things, but it's universal. And that means that basically archaea, any prokaryotes and eukaryotes, they read the genetic code the same way. For the most part, they read it the same way. So if I give a bacteria CAU, it's going to read it as histidine. If I put CAU in my own body, I'm going to read it, so eukaryotes, animal, right? I'm going to read it as histidine. So this is what we mean by universal. It's basically a single coding dictionary is used by viruses. Oh, I forgot viruses. Yes, viruses as well. Prokaryotes, archaea, and eukaryotes. There are some exceptions, but they're quite minor, especially when you compare them to the vastness of the organisms living in this world. I'm sorry I keep looking outside, but it's snowing, which is interesting. So beautiful. Okay, so going back to the triplet nature of the code. So with the experimental evidence, they saw that they needed a correct reading frame, so correct groupings, a contiguous, non-overlapping sequence of nucleotides in order for specified polypeptides to be produced. So they started messing around with this reading frame. Sometimes they basically started adding or deleting a couple of nucleotides. So when they did a gain or loss of 1 nucleotide, they saw that the reading frame was completely completely changed, and that the amino acid sequence that came about was completely different. They call this a frame shift mutation. When they saw if they gained or if they made the mRNA gain or lose 2 ribonucleotides, all of a sudden again the reading frame was completely changed, and we got completely new different reading frame and a different, I'm trying to move my screen, there we go. And a different amino acid sequence. So you can see here, reading frame 1, this is usually what's supposed to happen. You're supposed to have serine, leucine, threonine, alanine, etc., right? But then when we add one nucleotide, so gain one or even lose one, in this example, we're gaining, all of a sudden we're getting completely different amino acid sequence, valine, leucine, very different from the original, okay? When we gain, so in this case, we gain or lose 2, so AG, right? We again, serine, tyrosine, arginine, isoleucine, completely different from reading frame 1. But however, when they did this with 3 nucleotides, so they either gained 3 nucleotides on the mRNA transcript or they lost 3 nucleotides on the mRNA transcript, they reestablished that reading frame. And by reestablishing the reading frame, they got the correct amino acid sequence with just 1 amino acid extra, right? Like at the beginning or at the end, wherever they decided to put the new nucleotides. So the genetic code reads 3 nucleotides at a time in contiguous, non-overlapping linear manner. And this was basically what they realized from this experiment. And they used viruses for this experiment. Okay, so now let's actually look at how they figured out the genetic code, right? So the people who were really important in figuring out like the genetic code, so what code coded for which amino acid, were Nirenberg, Mate, and others. So like actually Ochai was one of them. They deciphered the code. So they used a couple of tools. First off, they were really, this is really cool because they were able to, they had in vitro protein synthesizing systems. So everything that they needed to synthesize a protein, right? So like the tRNA, the ribosome, everything, they had it in an in vitro cell-free test tube, pretty much. Okay, so it was there for their disposal. And the other thing that they did was that they radioactively tagged each amino acid so that each amino acid was different from the other. So they knew which ones were incorporated and which ones weren't. Then they also used this particular enzyme here called polynucleotide phosphorylase. Now, polynucleotide phosphorylase actually degrades, usually it digests and breaks down RNA into its individual building blocks. That's usually what happens. But if you put it in a test tube system, right? You put it in a test tube and you put in building blocks, so ribonucleotides, it actually puts the ribonucleotides together. It doesn't do so perfectly. It basically, so basically if you have 100% adenines, ribonucleotides, right? It'll make a poly A transcript just because that's all it has at its disposal. It basically incorporates depending on the concentration of the different nucleotides you have, the different ribonucleotides you give it. So the probability of inserting a ribonucleotide is proportional to the availability of that ribonucleotide relative to the availability of other ribonucleotides. And we'll get to a specific example later on. Okay, so this is what they had. They had the in vitro protein synthesizing machinery, right? And then they had this enzyme that made them able to make mRNA, which is great. So the first mRNA that they made were homopolymers. So homo means the same. This means that the RNA, each one that they made was completely identical. Like they had all of, they had the same ribonucleotide repeated over and over again. So the poly U had all U's, all like U, the transcript was all U's. Poly A, poly C, C. Unfortunately, they weren't able to make poly G's, so all guanines, because G's, I don't know if you remember from the telomere part from like the chapter 10, 11, I don't remember. But guanines are capable of actually forming single hydrogen bonds between themselves, and so they can actually bind together. And what they realized with poly G's is that the actual poly G would bend, and if you bend it, the mRNA doesn't work because remember we need it to be linear. So they basically added in a test tube to make the poly U, they added 100% uracil, okay, and then they added the polynucleotide phosphorylase, that enzyme, and then that enzyme just put all those uracils randomly together and they made the poly U. And then after that, they put it with the protein synthesis machinery and went ahead and saw which amino acid was actually incorporated, what chain, like what chain of amino acid of the same one amino acid would be made. And they saw with poly U, they made a chain of phenylalanines. So this is a particular amino acid. And so they concluded that UUU, this triplet right here, must code for phenylalanine. A similar approach was used for poly A. They realized that an AAA triplet coded for lysine and CCC coded for proline. So they were able to make homopolymers and figure out what each did. So these were the first mRNA like codes that they figured out. Then the next step was to make RNA heteropolymers, so mixed copolymers. So now we had 2 or more different ribonucleotides. So in order to make this, right, remember the phosphorylase, the polynucleotide phosphorylase just randomly incorporates. And so, but it incorporates depending on the composition of the amount of ribonucleotides available to them. So yeah, so we, what they did was they got, like in this example right here, let's see, I actually don't know the example. All right, so in this example, they added adenine and cytosine, but they added one adenine to every 5 cytosines, okay? So basically, yeah, there was like a 1 in 6 chance that adenine was going to be incorporated, okay? And then there was a 5 in 6 chance that cytosine was going to be incorporated. So I'm going to change the video, the camera. Okay, so so we know that there's a 1 in 6 chance. So there's like 1 adenine to every 5 cytosines. I guess I'm getting so many emails, I'm sorry. Okay, so what's the probability of getting an AAA? It's the first one up there, right? We know that there's a 1 in 6 chance that there's an adenine, okay? And so to incorporate an adenine, so the first one will be 1 in 6 chance, the next one will be 1 in 6, the other one will be 1 in 6. You remember the product rule, you multiply all that together. So there is a 0.4%, I'm just looking at the, I don't know if you can see my mouse, but at the table right here, there's a 0.4% chance that you will get the AAA triplet, okay? How about the chance for getting one with 3 C's? So C, C, C. We know that there's, to incorporate a C, that would be a 5 out of 6, right? Because it's 1 in 5 cytosine, 5 out of 6, 5 like out of 6. Multiply all that together, and that gives you a final percentage of 57.9. Then we have other ones, like maybe we have 1 C and 2 A's. Okay, that will give us, I'm just going to read off the table now, it happened about 6.9% of the time. But what does that mean? That could mean that our triplet code could be 2 A's and then a C, right? Like 1 A, a C, and then another A, C, and then 2 A's later, right? 2 days afterwards. So these are our possible our possible triplets. They didn't know which triplet would work. They just knew the composition, right, of the triplet. Okay, and so then once they did it all, they actually looked at the amino acids that were incorporated, and they looked at the percentage of the amino acids, and they compared it to the final percentage that each of these different possible compositions could bring about. So they saw that lysine was less than 1%, and so they were like, okay, lysine, and actually they even knew this from the other experiment. Lysine is probably encoded by AAA because that's less than 1%. They saw that something like yeah, something like 1A and 2 C's, whereas at 1A and 2 C's, that happened 12% of the time. So they were like, okay, probably it's 6, there's like 6.9 plus another 6.9. There's 2 triplets that code for threonine here, or something like that, right? So it was like likely that it was coded by one of these, of the 2C, or even like likely that it was coded by one of the 2C plus 1A right here, right? So they basically compared the percentages of the amino acids to the percentages of the codons and basically added things together. So for example, they had histidine. I can't see it because the Zoom stuff is in front of the table, so I'm just gonna write it out here. So I'm having difficulties, I'm sorry guys. So I have histidine, that happens 14% of the time. Now, if we look back at our final percentages of our codon compositions, we don't have something that's exactly 14, but it's probably maybe a 2C1A, right? Which is 2C1A, that's 6.9% of the time, or even a 1C plus 2A, that's another 6.9, plus another one, see how it's 2.3 of this, plus 2.3% of the time. So 7 plus 2, that equals about like 9, and so that's close to the 14%. So this is how they basically figured out, or like kind of try to figure out the possible codes, right? Yeah. So then the other thing that they did was another experiment they did. It was a triplet binding assay. So this was basically for specific codon assignments. So here they started realizing that they could make mRNA, they could synthesize mRNA and make and like actually know the order of the synthesis of the mRNA. So if they wanted to make an ACA, they could make it. But this synthesis of mRNA could only be very short, which was just fine because they could make 3. As long as they had 3 ribonucleotides, they had a triplet, right? So they made all these different triplets, and then with these triplets, they then, so it was like a specific triplet that they knew. They then looked at the amino acid that was incorporated with each triplet, and they saw that like they figured out which amino acid actually included it, like coded for it. So with this process, they were able to assign 50 out of the 64 triplets. 2 major conclusions were reached. The code is degenerate, so one amino acid can be specified by more than one triplet, and the code is unambiguous. Single triplet specifies only 1 amino acids, right? So this is kind of like what they got from, a couple of examples of what they got from the triplet binding assay. Right. And then finally, another technique that was actually developed by Corona, oh, that's funny, Corona, sorry, Corona, was he chemically synthesized long RNAs. So how he did that, again, you weren't able to synthesize, at that time, you weren't able to synthesize long RNAs, but he knew that he was able to synthesize short ones. So what he did is that he synthesized small ones, right, like so, and then afterwards, he joined the small ones together. So now you got a long one. He did this with di, right? So he made diribonucleotides and then added them all together. He made them with trinucleotides and then added them all together, and tetra, something like this, and then added them all together. So yeah, so he made these short repeating sequences, and then he enzymatically joined the short sequences together, which made long RNAs. So this was used for in vitro translation to determine the more codon assignments, and the data was all like upheld the triplet bind data that we got before from the triplet assays and the composition assignment data that we got in the second, in like one of the first, that first experiment. From these, he also basically deciphered the stop signals, right, and like all the other stuff. So he was able to do this because he knew exactly which, so if he had a dinucleotide UG, right, he knew that UG, you see it, don't pay attention to this purple and green part right now, but UG is always repeated. Now this, if it's always repeated, we get 2 possible like triplets from it, 2 possible codons. We get UGU and then GUG, then UGU, then GUG, over and over and over again. If we got a trinucleotide, all of a sudden we get 3 possible repeats. So if we get UUG, that's repeated over and over again, right? You could either have a triplet that's UUG or a triplet that's UGU, another triplet that's GUU. And so from this, you basically, yeah, and the tetranucleotides even more. It gives you like 4 possible triplets. And then, so he, through process of elimination, considering just like the other experiments, through process of elimination, he knew like which amino acid coded for which, like was encoded by which triplet. So from the dinucleotide UGU and GUG, I actually don't know what UGU stands for, guys. I'm sorry. Should go back to the, I'm not going to remember this. Do I not have it written down somewhere? I don't. Sorry. I'm going to forget the other ones as well. So, okay, so UGU codes for cysteine, right? And then we have GUG, which I didn't look up before, goodness me, but that codes for something else. I don't know. So actually, let's say amino acid 2. Okay, that codes for amino acid 2. So in the next experiment, he used trinucleotides where he got UUG, UGU, and then GUU. Okay, so let's pretend, so this is the first 2 and then the second 2. In this experiment, again, he got some more cysteine, and then he got, you know, I don't know, I'm making this up because I don't know what UUG stands for, leucine, and then this one is, I'm going to make it alanine. I'm making these up, guys. I don't want to go between the genetic code and this PowerPoint slide, right? So, but because he got cysteine here and he got cysteine in the first one, he could safely say that it was probably, and UGU was the only codon that was the same in both experiments. He could safely deduce that UGU gave you cysteine. And so that's how it was just basically through process of elimination that he essentially reaffirmed the identification of the triplets that had already been made, and he filled in the rest of the gaps that hadn't been made either. And he also actually figured out the termination codes as well. So that's it. We're going to start the next unit on chapter 12.4, but I'm going to stop here at section 12.3.